So much, Kim, and thanks to the NRF for sponsoring tonight's webinar so that we could all get together and talk about the exciting times ahead for Parkinson's disease research. And thank you to all of you for being here as well. So I'd like to begin by acknowledging the traditional owners of the land on which we meet today. I'm on Kaurna country, but I'd ask you to take a moment to pay your respects to the elders of the country that you find yourself on this evening. And I'd like to pay my respects to the elders past and present. Tonight, our goal is to talk about why, in my opinion, this is one of the most exciting times in history to be a Parkinson's disease researcher. I'd like to talk to you about some of the exciting developments that are happening in the field, and importantly, leave you with a message of hope about what's coming for Parkinson's disease, because I know many of you here tonight may be living with Parkinson's yourself, or you may be caring for someone who is. So the sort of way that we'll go about this tonight isn't a traditional lecture. I'm going to tell you a little bit about some of the science, but I'll try to keep it really clear and really practical, and leave lots of time for questions at the end because I really want this to be more of a conversation. And so first, I thought that I'd set the scene by talking about the big picture here at home in Australia. So up to 200,000 Australians, and some estimates suggest even higher, up to about 219,000 Australians, are living with Parkinson's disease right now. To put that in perspective, that's close to the population of Hobart. There's around 50 people diagnosed each day, which means that if we spend an hour together tonight, we'll be seeing, a number of people living with Parkinson's and receiving that news. And I think that one statistic that often surprises people is that Parkinson's is often thought about as a condition that affects older adults, but in fact, between 10 and 20% of diagnoses actually happen under the age of 65. So we call this young onset Parkinson's disease, and it affects more than 1,100 Australians every year. And one of the reasons that Parkinson's is receiving so much attention in the media and in the scientific literature right now, what's driving so much of the urgency in research is that there is a very large increase in the rise of Parkinson's expected in the coming decades. And you can see that on the graph here, showing that by 2034, it's estimated that there'll be a 79% projected rise. Now, this is driven in large part by our aging population, but also by environmental risk factors that can contribute to Parkinson's as well, which we'll talk about in a little bit. But this means that Parkinson's disease is now the fastest growing neurological condition in the world. It's growing even faster than Alzheimer's disease. And so that's why the research we're going to be looking at today matters so much, not just for the people in the room today, but for the next generation of people to be diagnosed with Parkinson's disease. And this is a very busy timeline, but the reason that I've included it is because I want to sort of show how things have been stagnant in Parkinson's disease research at various points in time. So it was first described by James Parkinson in 1817, and then it really wasn't until the 1960s where there was this big sort of paradigm shift in the field, which was the recognition that Parkinson's disease was associated with loss of cells that produce a chemical called dopamine in the middle of the brain, particularly in a structure called the substantia nigra. And this led fairly rapidly to the advent of dopamine replacement therapies like levodopa, which really revolutionized the treatment of Parkinson's disease and are still the gold standard treatment today. But you can see that since the 1960s, while there have been advances here and there, such as the advent of deep brain stimulation and increased understanding of some of the genetic factors in Parkinson's, we haven't had a major paradigm shift equivalent to what happened with that dopamine replacement therapy in the 1960s, I would argue until now. And I hope that by the time you leave tonight, I've convinced you that we're about to paradigm shift in Parkinson's by redefining Parkinson's disease based on its biological presentation, which I believe will greatly accelerate therapeutic development. So let's start big picture and talk about what actually happens in the brain in Parkinson's disease. So without getting weighed down in details, deep inside your brain, there's a small region, roughly the size of a peanut, and it's called the substantia nigra. And it gets its name because in the healthy brain, it looks quite dark. And you can see in the image here, you've got this strip of black And that's because these cells have a pigment in them called neuromelanin. And just like the melanin in your skin gives your skin its color, the neuromelanin in these dopamine neurons give them their color. And these pigmented cells make a chemical called dopamine. And dopamine is essentially dopamine is essentially the chemical that helps the brain to send smooth, coordinated movement signals to the body. Now, that's not the only thing it does. It also plays a role in cognitive function. It plays a role in eye movements, in mood, and even in addiction. But we'll focus mostly on its role in motor function. It makes walking or reaching for a cup of tea or smiling happen without you having to think about every single step. Now, in Parkinson's disease, for reasons that we don't quite understand, these dopamine-producing cells slowly stop working and they eventually start to die off. And you can see that here on the right-hand side. So whereas we had this really dark band on the left-hand side in an individual without Parkinson's, and there was lots of dopamine released from those healthy cells, you can see that in Parkinson's disease, there are many fewer of those cells and much less dopamine release as a consequence. And I think that one of the numbers that really shocks people is that by the time someone presents with the motor symptoms of Parkinson's, like tremor or stiffness or movement slowing down, studies have suggested that they've already lost between 60 and 80% of those dopamine-producing cells. And this is a really important point because it tells us two things. First, it tells us that the disease has been going on in the brain for decades, or at least years at the very least, before diagnosis. And second, it raises this idea that if we could detect Parkinson's disease earlier, before this catastrophic loss occurs, we'd have a much better chance of actually protecting those brain cells. And this is where modern research efforts come in. So the first research advance that I want to share with you tonight is research suggesting that Parkinson's disease may not begin in the brain for everyone. So recent research has supported the idea that Lewy body pathology, which is essentially the marker in the brain that we know is a hallmark of Parkinson's disease, as well as in other disorders that involve alpha-synuclein, it can begin in the brain, areas like the olfactory bulb, which is important for smell, or in the amygdala, which is a part of the brain important for emotion, or it can actually begin in the enteric nervous system of the gut. And so this has been used to propose some subtypes of Parkinson's based on either brain-first Parkinson's disease or body-first Parkinson's disease, depending on where that pathology first arises. And this is important because it can be associated with different disease presentations, at least initially. So, for example, in people who have body-first Parkinson's disease, where you see that first pathology in the gut, you can actually see more sleep disturbances like REM sleep behavior disorder and more autonomic changes like constipation and orthostatic hypotension, which is the low blood pressure drops that can happen when someone stands up. And these are quite common in body-first Parkinson's, but we don't really see those in brain-first Parkinson's disease. And it's been suggested that these different presentations can help to differentiate Parkinson's disease from other disorders like dementia with Lewy bodies, because we know that in Parkinson's disease, for example, we tend to see more of a brain-first presentation. About 2/3 of cases are brain-first, whereas in dementia with Lewy bodies, we tend to see more of a body-first presentation. And you may be wondering, isn't Parkinson's? Who cares where it starts? Well, the reason that this is important is because if we understand where Parkinson's disease begins and when it begins, this is really key for helping to inform risk and progression, and also can help give insight into clinically distinct subtypes and phenotypes, which can help do things like predict disease trajectory and inform more personalized care and management strategies. So this is an idea that's still very much in its hypothesis phase. So it's now being tested with molecular biomarkers, it's being tested with neuroimaging, it's being tested in large clinical samples in order to understand how this hypothesis, whether or not it holds true. So stay tuned, but it's definitely something that's changing the way that conceptualization of Parkinson's happens. And also in this same vein of this early detection, The second research advancement I wanted to share with you is the idea that I've already briefly touched on, which is that Parkinson's disease begins years or decades prior to motor symptom onset. So when we look at when that clinical presentation of Parkinson's happens, which is where I'm indicating right here, right near this dark orange in the graph, you can see that there's this whole progression to the left of it that's happening in the years and decades before. And again, this is really critical because by the time we reach this dark orange point, by the time we reach this point where motor symptoms are manifesting, we already have this really significant dopamine neuron loss. So if we could instead predict Parkinson's disease and identify Parkinson's disease here, or even better, here or here, we could actually track people and help to hopefully protect those brain cells before they're lost. Now, one of the things that makes this a little bit tricky is that the symptoms that are associated with what we call prodromal Parkinson's, which are the symptoms that happen before the motor symptoms, can be really quite varied. Two of the most common that are talked about are loss of sense of smell and sleep disturbances, like REM sleep behavior disorder. And of those, REM sleep behavior disorder really has the strongest evidence for later progression to Parkinson's. Now, if you don't know what REM sleep behavior disorder is, REM is the period of sleep in which we dream. REM stands for rapid eye movement. And what usually happens when we dream is that our muscles are deeply relaxed so that we don't act out our dreams. But what happens in people with REM sleep behavior disorder is that during that REM period, their muscles are quite rigid. They can thrash around, they can act out their dreams. If any of you here tonight are living with Parkinson's disease, your partner may have said to you, even before your diagnosis, oh, you're keeping me up all night, you're just thrashing around, you're yelling out in your sleep. And some of these symptoms can be associated with this REM sleep behavior disorder. But there can be other symptoms as well. These can include autonomic changes like changes in constipation, or sexual function. It can include mood changes like depression. It can include blood pressure changes. And one of the things that makes this tricky is that these can be relatively nonspecific. So sometimes when people go to their GP, they just say, oh, you're getting older. Excuse me. These are just part of normal aging. We need to get better about educating clinicians that when we see a constellation of these symptoms getting worse over time, we may want to have some enhanced monitoring and follow-up. So these symptoms can be used to calculate what's called a prodromal score. So the Movement Disorder Society has research criteria that we can actually use to calculate someone's prodromal score, which can help to inform their risk prediction. And again, this is really critical because if we can track prodromal symptom presentation in known at-risk cohorts, we could potentially detect Parkinson's disease earlier. So then this raises the really critical question of what drives Parkinson's disease risk. For most people, Parkinson's disease isn't one cause, it's a whole mixture of factors. And this is something that I often get asked when I'm talking to people in the community with Parkinson's. They ask me, why me? How did I get Parkinson's? Where did this come from? And often people will ask me, I have Parkinson's disease. What does this mean for my children? What does this mean for my family members? Well, one of the things that I want to really emphasize is that Parkinson's disease, when we look at pure genetic causes, they only occur in a relatively small number of people, about 5 to 10%. So 5 to 10% of people with Parkinson's have a form of the disease that's directly caused by a genetic mutation. And in fact, most people with Parkinson's disease don't have a family history of it. So some statistics have suggested that it's anywhere from sort of 10 to 15% of people where there's a clear family history and a clear genetic cause. So what that means is that if your parent or grandparent didn't have it, it doesn't necessarily protect you from developing Parkinson's. And if your parent or grandparent did have Parkinson's, it doesn't mean that you yourself will get it as well. Now, for the vast majority of people instead, the biggest predictor is actually environmental and lifestyle factors. These include things like the chemicals you've been exposed to over a lifetime, so pesticides, organic solvents, head injuries, especially from work or sport. So there's been a lot of studies, particularly in US veterans, suggesting that people who've had even a concussion are at elevated risk of Parkinson's, whereas people who've had more severe head injuries, that risk can increase to about 80%. Now, I do want to qualify that by saying that the vast majority of people who have concussions and head injuries do not develop Parkinson's disease. We just know that having such a history can elevate your risk. So definitely, if you've had a concussion, I don't want you to think, oh my gosh, there it is, there's the explanation. We just talk about things that raise relative risk. Now, pesticides and head injuries have definitely received the most attention in the literature, but there's a growing focus on other factors, like the role of air pollution and the little particles that are found in air pollution, and even the role of microplastics. So there's some really interesting work going on here in Australia looking at microplastics. Now, in addition to those sort of environmental factors, we also know that male sex makes developing Parkinson's disease more common. So Parkinson's disease is about 1.5 times more common in males. And of all the risk factors that have been studied, age itself is the biggest risk factor. And so that's why, given that we have such a rapidly aging population, it's really critical that we invest in Parkinson's disease research now, that we try to increase our understanding of Parkinson's disease now, so that as our population ages, we'll be equipped to cope with that. Now, the one good thing about environmental factors is that in principle, they can be changed. So when we talk about lifestyle factors like diet and exercise and heart-related factors, these are all things that lifestyle modifications can help with. And in fact, we're seeing a lot of research come out of the dementia literature suggesting that if we target those sorts of modifiable risk factors, we can dramatically decrease the number of cases of dementia. And while the literature on Parkinson's is smaller, it's supporting similar decreases with lifestyle factors. And so then this leads into our third research advancement that I wanted to share, which is growing recognition of the role of something called the exposome. So traditionally, environmental exposures in Parkinson's have been understudied, and there's a lot of reasons for that. It can be really difficult to capture someone's environmental exposure because you're asking people to reflect back on their life and to think about, all the exposures that they may or may not have had. But there are now more objective ways to look at that, using things like agricultural data and weather data, soil analysis, in order to really more objectively quantify those measures. And this has led to what's called the exposome framework. And what the exposome is it's all those things you've been exposed to throughout your lifetime. So it's these general external exposures like pesticides and air pollution. There's your individual external exposures like your diet, your history of smoking, your history of exercise. And then there's your internal exposome, which is all the things that your biology, essentially your underlying biology. And the idea is that we can take this framework of these different components and take what's called a biography to biology approach. And what that means is that we may be able to use machine learning techniques to calculate what are called poly-exposure scores in order to actually quantify environmental risk profiles. And this is really critical because a score like this could help us to understand the contribution of lifetime exposures to Parkinson's risk and how these interact with things like genetic factors. And it's understanding this that's really key for prevention and prediction of Parkinson's. So this is what my lab focuses on quite a lot. We're really interested in the idea of how do these lifetime exposures, things like head injury, things like pesticide exposure, increase someone's risk for Parkinson's, or even change the trajectory of Parkinson's for those who currently have established Parkinson's. Now, it's probably not surprising that if we're going to talk about lifetime exposures, if we're going to talk about genetic risk factors, we need really large-scale studies in people with Parkinson's disease and people in at-risk populations in order to try to tease these factors apart. And so the next research advancement I want to tell you about is a study that's going on right here in Australia, which is called the Australian Parkinson's Genetics Study. Now, I myself am not directly involved with this study, but I've been following their results with a lot of interest because it's the largest ongoing active Parkinson's study in the world. And they've just reported data from nearly 11,000 Australians with Parkinson's disease. So this is a nationwide study of Australians with Parkinson's, and essentially what they did is they contacted people by mail based on their Medicare data, and they asked them to complete a detailed questionnaire asking them about their experience with Parkinson's disease, their lifestyle factors, their demographics, and they also asked them to provide a saliva sample that could be used for genetic testing. And participants were able to join from home anywhere in the country, so they could join by mail. Some testing was done online. And the average age of participants was 71 years old. The study reported that people first noticed their symptoms of Parkinson's at 64. And the overall goal of this is to find the causes of Parkinson's disease in order to try to understand how genes and environment may lead to Parkinson's disease and to try to speed up the development of new treatments, especially personalized treatments that may be more tailored to each individual person. So what has APGS already found? So I'll take you through some of the findings that I think are really striking. And the first is that it told us that non-motor symptoms are really quite common in Parkinson's disease. And while the literature has widely reported this, it's good to have that confirmation in a large-scale sample, particularly here in Australia. So we saw that, 52% of individuals with Parkinson's had experienced loss of smell. 65% had changes in memory, 66% pain and dizziness, and a really incredible 96% of people with Parkinson's reported sleep disturbances. So this really tells us that the motor symptoms of Parkinson's are just the tip of the iceberg, and that non-motor symptoms are actually really significantly affecting people. And these largely drive quality of life for individuals. It also gave us a better picture of risk factors. So it told us that 1 in 4 people with in this particular cohort had a family history of Parkinson's, which is a bit higher than what we've seen reported in the literature. 36% of people had a history of pesticide exposure. A third of people worked in high-risk occupations like agriculture or petrochemicals. And 16% of people had a prior history of head injury. And so it really helped to clarify what are the risk factors that may lead to Parkinson's and provide additional support for those environmental factors we talked about a moment ago. And I think one of the most interesting things that APGS revealed in this first observational data release is something that's really understudied in the field, which is how Parkinson's disease may affect men and women differently. We know it's more common in men, but actually women, at least in this cohort, were younger at time of symptom onset, and they were younger when they received their Parkinson's diagnosis. They also experienced symptoms differently. They were more likely to experience pain and falls, whereas men were more likely to experience memory changes and impulsivity. So it gives a lot of insight into the fact that maybe clinical management needs to be different between men and women, that there may not be a one-size-fits-all approach that's appropriate here. But some caveats to keep in mind. This was the first release of the data. It's purely observational at this time. And although they sent out surveys to many individuals, they only had about a 6% response rate. So it may be a biased sample and not fully representative of Parkinson's disease as a whole. Additionally, the people responding, 93% of them were of European ancestry. So we know that other ancestry groups are underrepresented in this sample. And because of the male nature of it, they weren't able to collect objective measures of things like motor function or cognitive function. They had to rely on self-report, which we know can have issues. People can sometimes not recall things accurately, or they may not, tell you the full story. And currently, there's no controls to compare this to. So we don't know how this particular cohort of people with Parkinson's compare to age and sex matched controls. So people of similar age and who are, male or female in the non-Parkinson's disease population. So I think there's some really interesting advancements, but it's a watch this space because the study's ongoing and they're going to continue to report data. So hopefully I've convinced you that large-scale studies have value, but in addition to these large-scale studies, we also need better biomarkers for Parkinson's disease. And this is where things get really exciting, and this is arguably the biggest change in Parkinson's disease of the last, 200 years. Since Dr. James Parkinson first described the condition back in 1817, the way Parkinson's disease is diagnosed has barely changed. A doctor looks for the classic motor symptoms, resting tremors, stiffness of movement, slowness of movement, balance problems, and uses that motor symptom presentation in order to make a clinical diagnosis. But there's some very significant sort of caveats with that and limitations, which is that there have to be visible and often advanced motor signs for someone to actually go to their GP and receive a referral to a specialist who can make that diagnosis. And by then, most dopamine cells, as we've already talked about, are already lost. This approach also doesn't acknowledge the heterogeneity of Parkinson's disease. It can look quite different between different individuals. People don't always have the same motor profiles. And it doesn't acknowledge the non-motor symptoms, and it also doesn't acknowledge the underlying biology. And I always find this statistic quite scary, which is that misdiagnosis happens in about 1 in 5 cases of Parkinson's disease. And this can be sort of diagnosing Parkinson's disease for something else, one of the Parkinson's plus syndromes, for example. And this can really delay people being able to get appropriate treatment. It can shake confidence in the medical system. So it's a really big problem in the field. But the new way that the Movement Disorder Society is really, beginning to push for is to integrate biology and to diagnose Parkinson's disease in light of its biology. And this can involve many different measures. It can involve things like detecting misfolded alpha-synuclein in body fluids. There may be neuroimaging using MRI or PET scans to look at dopamine loss or loss within certain brain regions. It can involve genetic analysis to look for known gene variants. And this is a really powerful combination because it can potentially detect the disease years before those motor symptoms occur. Now, this is still very much in its research framework stages, and the field is trying to understand how to appropriately integrate this into practice and when it's appropriate to do so based on confidence in the biomarkers that are currently available. But it's really advancing quickly, and it's just been in the past year and a couple months where this conversation has just taken off internationally. And this has been driven in large part by 2 frameworks for the biological staging of Parkinson's, which have been really influential. Now, before we get more into biomarkers, I just want to take a really quick time out because we've talked about dopamine, but I've made a couple mentions to this protein called alpha. I haven't introduced a small protein that has a really big role in our brain cells in healthy.
Lindsay, sorry to interrupt. I just want to let you know the audio's just gotten a little bit distorted from my end. So just in case that's happening for anyone else, maybe we'll just pause it for a second and see if the connection can catch up.
Like a heads up when I should start again. How's that? Is that a little bit better?
There we go. Yeah, that's come back in.
Okay.
I just thought I'd let everyone else know as well. If there's any issues, please pop them in the chat if you can't hear or it all looks good from my end. But yeah, just let us know if there's any issues. And please pop your questions in the chat. No worries, that's much better, Lizzie. Sorry about that.
That's okay. No, thank you for the heads up. I'll just very quickly go over that again, just in case it was missed. So what I said was that we'd already talked about the role of dopamine in Parkinson's disease, but that what we hadn't talked about, even though I'd made some allusions to it, is the role of alpha-synuclein. So alpha-synuclein is a protein that we find in the brain, and alpha-synuclein in healthy brain cells helps those brain cells to talk to one another. In Parkinson's disease, for reasons that we don't understand quite yet, this protein starts to misfold. It changes shape, a little bit like crumpling up a piece of paper. And once it misfolds, it can no longer do its normal job. And worse, it starts to cause healthy alpha-synuclein nearby it to misfold as well. It's kind of like if you have one bad apple and then it spoils the whole barrel. If you have one, misfolded alpha-synuclein, then it can act as a template to misfold all the other healthy alpha-synuclein. And what ends up happening is that these alpha-synuclein, this misfolded alpha-synuclein clumps up, and don't worry about the names of the different clumps, but eventually it clumps together into this structure called a Lewy body. And Lewy bodies were actually first described over 100 years ago, but we now know that they contain mostly alpha-synuclein. And so a lot of research today is focused on how can we measure alpha-synuclein in people with Parkinson's or at-risk individuals, and how can we potentially target misfolding alpha-synuclein to treat Parkinson's disease and actually, potentially slow or stop Parkinson's disease rather than just treating the symptoms. And that's a huge shift in mindset from where we were even 10 years ago. So it really matters because if we can detect, slow, or prevent the misfolding and aggregation of alpha-synuclein, we may be able to change the course of the disease itself. And so our next research advancement that we'll talk about is a breakthrough biomarker test. And this happened really about a year ago for the first time, which is that for the first time, we can detect alpha-synuclein in living individuals with Parkinson's disease directly. And this is called the alpha-synuclein seed amplification assay, or SAA for short. And here's sort of roughly how it works without getting too much into detail. So a clinician takes a small sample. Right now, it's usually cerebral spinal fluid, which is the fluid that we find in our brain and spinal cord, which you can collect with a lumbar puncture. But increasingly, people are looking at skin biopsies, blood samples. They're even looking at tears and whether or not you can measure alpha-synuclein in these more accessible and less invasive biofluids. And what you do with that sample from the patient or from the individual that you suspect may have Parkinson's disease is that you add it to a solution that has healthy, non-misfolded alpha-synuclein in it, and then you track the misfolding. Because remember when I gave that example of like if you have one bad apple in the barrel, it spoils the whole barrel of apples? Well, if you have the patient sample that contains misfolded alpha-synuclein, it will cause that healthy, unmisfolded alpha-synuclein to misfold. And so we say that it seeds the misfolding of alpha-synuclein. And then you can actually use a fluorescent signal to measure whether or not you get that misfolding. And the accuracy of this test is remarkable. So in published studies, it can correctly identify people with Parkinson's about 96% of the time. And it can rule it out in people who don't have Parkinson's about 92% of the time. And that's almost as good as it gets for medical tests. So the US Food and Drug Administration, or FDA, has issued support for using this test in clinical trials. And many labs are rolling it out for research. And we're starting to see, discussions about whether or not this will enter into clinical practice. And what I think is most exciting of all is that, yes, it can detect this misfolding in people with Parkinson's, but also in people who don't yet have motor symptoms. So for example, if you look at people with REM sleep behavior disorder, which we talked about before as one of the prodromal symptoms that can precede Parkinson's disease by years, they also have positive alpha-synuclein seed amplification. So this opens the door to a biomarker that can let us see risk of Parkinson's disease before those motor symptoms. And this is huge for the field. It's really hard to oversell how significant this is. And It's not just alpha-synuclein that's being looked at because Movement Disorder Society and other researchers have really cautioned that we can't just look at alpha-synuclein in isolation because Parkinson's disease is heterogeneous and there's a number of biomarkers that are implicated. So also labs are giving us an improved understanding of Parkinson's disease biology. So this was a big story that came out last year, which is that researchers at WEHI have discovered what human pink-1 proteins look like. So for a long time, we've known about mutations in the gene that codes for this protein in Parkinson's disease, but no one had ever been able to visualize it before, or visualize how it relates to something called mitochondria, which are within your cells and help to power those cells. They're like the energy centers for the cell. And what we know now from this research, which came out just last year, is that pink-1, that protein, can sense damage in those energy centers, and it can attach to them and basically tag those mitochondria to get recycled, and this helps to maintain normal cell function. And so we know that this can become abnormal in Parkinson's disease. So having this understanding of the structure of pink-1, which you can see here, is really key because it may help us to develop new drugs for this target and give greater insight into the biology of Parkinson's disease. So we've talked a lot about research advancements, but let's talk about what all of these advances in the science are actually delivering. So levodopa and dopamine replacement therapy, they've been around since the 1960s, still remarkably effective, still the gold standard, but they treat symptoms. They don't modify the disease. Instead, what we're starting to see is movement in the field to actually develop therapeutics that can modify the disease. So one of this is actually surgical advances. So they don't directly modify the disease, but many of you would be familiar with deep brain stimulation. If you're not, this is essentially like a pacemaker for your brain. So just like a pacemaker for your heart helps to set a rhythm based on stimulation parameters. Same thing with deep brain stimulation. There's an electrode and it delivers a constant electrical current to the brain. But right now what happens is that if you need those stimulation parameters adjusted, it has to be done manually by a clinician. Last year, the FDA approved a new technology using smart electrodes that can actually allow the electrode to make real-time adjustments for stimulation parameters on its own, which means that there aren't delays in adjustment between clinical visits, and it can really respond to a person's needs at a given moment in time. So this is a really exciting advancement in the field. Also, just last year, we saw the approval of various infusion therapies. So these include things like Vyalev, which is subcutaneous levodopa, or Onopgo, which is continuous apomorphine. And these can help to smooth out those on-off motor fluctuations. So it's not a cure, but it is a real, quality of life advance in helping to smooth those transitions. And then there's the synuclein therapies, the anti-synuclein targeting therapies. So antibodies like prasinezab and small molecule aggregation inhibitors are moving through phase 2 and 3 trials. Several Australian sites are participating in this, so it's really exciting to see whether or not directly targeting alpha-synuclein may be beneficial. And stem cell therapies. So some of you may have followed about a month ago or so, but dopamine-producing neurons can actually be derived from pluripotent stem cells that get implanted into patients. And based on early trial results, a particular stem cell therapy at Kyoto University has actually received limited and conditional approval in Japan. So this is a therapy that's really rapidly advancing and is really potentially exciting to actually help to restore lost neurons. Gene-targeted drugs that target the specific genetic alterations we see in Parkinson's, like LRRK2 inhibitors and GBA-directed therapies, may help to really genuinely modify the disease by targeting biology of specific genetic subtypes. And finally, new targets beyond alpha-synuclein or dopamine replacement are currently in trial. And these can include things like targets for neuroinflammation, which is something that my lab works on, targets for the mitochondrial dysfunction that we just talked about a moment ago with pink-1. And there's also a lot of work being supported around drug repurposing. And the goal of this is really to help slow or stop neurodegeneration. So for the first time, the question is really shifting from how do we manage symptoms to how do we slow, stop, or even prevent the disease itself. And so far we've talked about the science, but science alone doesn't change lives. It has to be matched by policy, by services, and support. And so I wanted to share with you some really exciting news for Australia. So just a couple weeks ago, in late March, the Australian federal government and the National Parkinson's Alliance launched Australia's very first National Parkinson's Action Plan. And the National Parkinson's Alliance is a fairly new body. It aims to bring together researchers and Parkinson's organizations and clinicians from across every state. And they have received federal funding commitment in order to get that plan underway. And essentially what they have suggested is that there be 6 outcomes that we focus on to try to alter the course of Parkinson's disease here in Australia, focused on tackling stigma in order to change how Parkinson's is understood by increasing awareness and having more accurate information about the disease and its treatment, strengthening prevention by delaying onset and improving the understanding of risk and disease progression, improving diagnosis, so faster recognition, more empathetic delivery of diagnoses when they do occur, making sure that you spend time with an individual to help them through the diagnosis and coming to terms with the diagnosis, quicker access to specialists and allied health, which then ties into better care, which is access to evidence-based treatments and supports and multidisciplinary teams, regardless of if you live in metro areas. And to support that, helping to build capacity and capability of the workforce by upskilling doctors, nurses, aged care staff, disability staff, to really be able to appropriately recognize and manage Parkinson's disease. And finally, maximizing research impact by improving data, maximizing the impact of research, making sure that it's addressing the priorities of the Parkinson's community. And so this is really exciting. I can't wait to see how this is put into action. I know that organizations like the NRF are already really working across these pillars with events like tonight to try to increase awareness and education, but also investing in research across the continuum from risk identification to new treatment development. So really exciting. So the take home I really want to leave you with is that there's a real reason for hope. We can see Parkinson's earlier than ever. We know more about what causes it, and we're targeting the disease itself, not just its symptoms. So if you're living with Parkinson's or a loved one is living with Parkinson's, know that you are part of a community and that community is moving forward. So thanks for joining us tonight. I'd just like to acknowledge my lab and some of my major collaborators, funding bodies, and our community partner, Hospital Research Foundation Parkinson's, who plays a critical role in helping us with our recruitment of participants. NRF helps us with both recruitment and funding, and various other funding bodies like the MRFF, NHMRC, and James and Diana Ramsey Foundation also play a key role in our research. So with that, I'd like to turn it over to questions. And I understand, Kim, that you're going to be asking me them.
That's right. Can you hear me okay? I'll jump back on.
Yes.
So yes, I will be asking the questions. Now, I just wanted to flag because I haven't actually seen many come through the actual Q&A or the chat, which I've enabled. So if anyone has any issues with that, please let me know. And if you would prefer not to type the question, just raise your hand. There should be an option just above the screen where you could see Lyndsey's slides, and I will unmute you so that you can ask a question. However, we have had quite a few sent through when people registered, so I thought I'll just make my way down the list, and yeah, you can answer them as we go. So the first one you did already touch on a bit in your presentation, but it is, how is Parkinson's disease diagnosed currently, and what has your research identified as some of the earliest signs and symptoms that can help with earlier diagnosis? So maybe if you just wanted to do a little summary of that. I know you touched on it in the presentation, but maybe, yeah.
For sure. So as I mentioned, Parkinson's disease diagnosis currently has not advanced much in the last 200 years-ish, it's very much still a diagnosis of exclusion. So what people will often experience when they go through a Parkinson's diagnosis is that their motor symptoms will be assessed, and then they will receive blood tests to rule out sort of other causes, like infections, for example, or alternate causes. Neuroimaging may be part of that as well, to rule out things like a brain tumor or a stroke. And then often the sort of third thing that's done is that health professionals will look at response to dopamine replacement therapies like levodopa. And if the person's motor symptoms respond to that, then they say, okay, well, it's Parkinson's. That's obviously not highly specific. And so increasingly, we're seeing some clinicians incorporate some other biomarkers. These may be neuroimaging biomarkers like nigrosome imaging, in some cases PET scans like F-DOPA PET. We're not really at the stage where specific blood-based biomarker tests are being used, but this is where the field is rapidly going with those seed amplification assays that I mentioned before. And so I think what my research has really suggested is that using those clinical symptoms alone is not enough. So we've looked across hundreds of people with Parkinson's disease. We've looked at the presentation and progression of their motor symptoms, but also their non-motor symptoms like cognitive function or mood dysfunction, like related to depression or anxiety. And what we found is that when you incorporate biomarkers into that process of tracking those symptoms using both advanced statistics, but then also replicating it with machine learning, we can significantly improve prediction of someone's journey with Parkinson's disease for both their motor and their non-motor symptoms. So we're really passionate about trying to advocate for the incorporation of evidence-based biomarkers into clinical practice for these reasons, to try to avoid misdiagnosis and help inform people's individual trajectories as well.
Fantastic, thank you. Very informative. So the next one I have is from a Parkinson's MSAC patient. Now, I'm actually not familiar with that term, but I imagine you probably are. So it was really just a bit of a better understanding of that particular subtype of Parkinson's. So it's got here better understanding of MSAC. So are you able to touch on that at all?
Well, so one of the frustrating things about Parkinson's disease is that it's one member of a larger family of related disorders. And so this can include things like PSP, which stands for progressive supranuclear palsy, MSA, multiple systems atrophy. There's a variety of others as well, like dementia with Lewy bodies is related as well. And these can have overlapping symptoms with Parkinson's, but they can actually be quite different in terms of their brain presentation. They can be quite different in terms of their trajectories. And I think one of the things that's limited really a deeper understanding of some of these Parkinson's plus syndromes is simply patient numbers. So, we've really been recruiting for a very long time for our Parkinson's cohort, and even that has been a tremendous uphill battle. And so, recruiting in these Parkinson's plus cases, where often there are a much smaller pool of people to recruit from, can really limit research in that area.
And I should mention that as well. So that's the FIND TBI? Is that the one that you're referring to? Yeah, so we should mention that to anyone on the line if you haven't heard of it. Yeah, it is still open for participant recruitment. So looking for either patients who have Parkinson's or have had a history of a traumatic brain injury, I believe. Is that correct, Lyndsey? And then, yeah, you can reach out to Lyndsey or the NRF and we can, link you to the people running that trial. Because that is currently ongoing. So definitely reach out if you're interested.
And if you have Parkinson's and you don't have a prior history of TBI, that's totally fine. You're still more than welcome to participate. So we run our Find TBI and our Play Your Part for Parkinson's studies in parallel with one another. We're really interested in that link between head injury and Parkinson's that I mentioned before.
Excellent, thank you. So the next question we had was, I'm interested in hearing discussion and research outcomes relating to incontinence.
Yeah, so this is a bit outside my own area. And so it's definitely, I think one of the things in general that I can say is that the non-motor symptoms are receiving quite a bit more attention. And so Traditionally, there's been a lot of focus on Parkinson's as a motor disorder. Now it's really well recognized that Parkinson's disease is not a motor disorder, or not solely a motor disorder, that we talk about the Parkinson's iceberg and all of the non-motor symptoms. Incontinence and urinary changes can be part of that. They can be part of the prodromal symptom profile. In terms of specific therapies in development for them, I'm not aware of the specific therapies, but I do know that in general, investment in non-motor symptoms ranging from pain to cognitive dysfunction to the gut and urinary changes we see in Parkinson's is really increasing, and recognition and awareness is really increasing as well. And a great place to see therapies that are currently being invested in and that are in development is actually through the Michael J. Fox Foundation website. So if you go to that website, you can often see the various motor and non-motor add-on therapies in development.
Great, thank you. So the next one we had was actually about the relationship between Parkinson's and exercise. So someone has said high aerobic intensity exercise is often said to slow or even reverse the progression of Parkinson's. Is there any firm scientific evidence to support these claims, e.g. double-blind random controlled trials with strong p-values or significance thresholds? How much exercise is required? So heart rate, duration, frequency. I don't know if you know
One
anything in
of the
that
things that
area.
makes a double-blind study with exercise difficult is that you know if you're exercising or not. And so it can be really difficult to have a true double-blind study. And this is the case with a lot of sort of non-pharmacological interventions. It can be really difficult to blind them appropriately. And so we talk about what are called passive controls, which is where people just have their sort of everyday normal activity. And there are studies that compare physical activity to normal activity. There's also active controls where you may compare two different types of exercise interventions to each other, one that you think will be more effective than the other. Some studies incorporate all of this. The evidence for exercise in Parkinson's, the evidence for exercise in neurodegenerative disease in general is pretty darn good. And this isn't necessarily just aerobic exercise. This also includes other forms of exercise as well. And in fact, exercise is now well recognized as sort of one of those physical activities recognized as one of those modifiable risk factors for dementia to help prevent dementia cases. And we know that one of the things that exercise does on the neural level is it helps to promote neuroplasticity. It's neuroprotective. It helps to stimulate growth factor production. So it's more than just feeling good. It actually is having changes on a neural level. So if you have Parkinson's, or if you don't for that matter, get moving. And I think that we're seeing lots of interventions for people with Parkinson's around things like Dance for Parkinson's, etc., which really has dual benefits. There's the physical activity component, and then there's also the social activity, because we know that social activity can also be incredibly beneficial.
Fantastic, thank you. We've had something come through the Q&A chat. So this is from Kevin. He said, is the Australian research study still open? And so that would be what we spoke about before. And do they need additional participants? So I feel like we've
Um,
answered that a little bit. But I also did want to ask, are there any other ongoing Australian studies that you know about, or trials that are like
yes, there are.
recruiting? Yeah.
Yeah, so the Australian Parkinson's Genetics Study, I believe, is still recruiting. And so if you sort of go online, you'll find their website and they'll have more information on that. I'm not sure, because I'm not involved with that study, I'm not sure exactly what the mechanism for expressing interest is. I know the initial study was done through mail outs, so I don't know if there's a way for people to contact them. Our studies are recruiting. We're looking, as we said, for people with Parkinson's. Please reach out to myself or to the NRF. We would love to have you participate. And then there's also a number of other ongoing studies in different states and at different times. So it depends on whether or not people are recruiting nationally or not. And one of the best ways to know that is by going to look at the various sort of Parkinson's support groups, places like Hospital Research Foundation Parkinson's, places like the NRF here in South Australia, because they will help to disseminate that information when they know about studies that are happening through their social media. They do it through their newsletters. Also, looking for things like National Parkinson's Alliance and Parkinson's Australia, they all Shake It Up Foundation as well. They'll often also advertise studies that are going on. So, particularly, I believe Shake It Up has a page of ongoing studies that are happening. Parkinson's Australia also has a Parkinson's Research in Australia page. So, have a look at all of those resources if you're interested in participating, or contact me. And also, I should say that. Well, maybe I can't say this. There are some applications that are going in by large national groups for studies in Parkinson's that, if successful, would recruit nationally on a very large scale.
Oh, that's great. Bit of inside research, Gus. We'll all keep our fingers crossed for that. That's very exciting. Um, Now, I've got a couple of questions. They're a little bit probably more clinical treatment focused, but I thought I'll put them out there because other people might be interested, and then yeah, you can respond the best you can. So one of them actually came through prior to the webinar. I just won't put in any identifying information, but basically it was a question about the common Parkinson's medications, so levodopa, I won't be able to say it, but benzodiazide and entacapone. So many people would probably be aware of those. And it was just about deteriorating mobility. And is there, essentially, do you need to maintain those medications once the mobility deteriorates? And is there sort of a point in time that perhaps you decide to discontinue those medications if they're not really showing much benefit? Or are you better off just maintaining them regardless of that level of function?
So I'm not a clinician, so I'm a researcher, and so I'm not qualified to offer medical advice. So I can offer sort of the research perspective from the literature, but any conversations about, treatment and medication should always be had with your healthcare professional. What I would say from the research literature point of view is that there's evidence that Parkinson's disease medications like certain symptoms of Parkinson's can still be responsive to medication, even in quite late stage Parkinson's. This can include, for example, things like swallowing difficulties. And so the general advice from the literature is to continue to maintain medications even in late stage Parkinson's to help with, comfort levels for the individual. But again, any specific questions around that, really make sure you talk to your healthcare professionals because they're there to help work with you in partnership to manage your medications.
Thank you. Um, and we had someone else ask that it was suggested to them by a nurse that it might be a good idea to register with a geriatrician. They see a very helpful neurologist at the moment and just wondered if it was an idea worth considering. Oh, I see. Sorry. So they weren't particularly happy. They had a neurologist in London that they weren't particularly happy with, and then switched to a geriatrician and were much happier. So is it acceptable for them to communicate with each other regarding care?
Um,
Um,
So I think one thing that the National Parkinson's Alliance and the National Action Plan has really emphasized is the need for coordinated care in Parkinson's. So having multiple different members of your medical team and having good communication between them. This may include, allied health professionals you're working with, like speech pathologists or physiotherapists, nurses, people working with you, whether it be a neurologist or a geriatrician. Now, in practice, I think that that coordination between different health professionals can be tricky. And so I'm hoping that National Parkinson's Action Plan will provide some actionable steps to improve that. But I think that that's still in progress. Now, that being said, I know individuals who are managed by geriatricians. I know people who are managed by neurologists. Each have, wide knowledge about Parkinson's, and I know that both get great care. So I think it really can depend on the individual and their fit. And I think that's true even within specialties. So some people may just really find that the bedside manner of a particular neurologist resonates with them or doesn't. And so I think my message would be, if you're not happy with the care that you're receiving, it's your right to seek out different care. And you should, because Parkinson's can be a long journey. It can sometimes be a very overwhelming and lonely journey. But remember, like, on this slide here, you are part of a community. So reach out on those support systems.
Thank you. That's really good intel. So we have another one about focused ultrasound. So just wondering if you had any knowledge or could provide any more detail about focused ultrasound for the treatment of motor symptoms of Parkinson's.
Yes, so that was another one of it almost went on my slide and I ran out of room in the box with the therapies. So focused ultrasound is another one that has received some approval. It's really advancing through trials. So that's one that I would watch. So similar to those advances that we're seeing with the more adaptive deep brain stimulation, focused ultrasound is also really progressing through trials. And so that would be a space to watch.
And does it seem like it's getting quite positive results? Like, is there any literature around, it's showing some positive effect?
Yeah, so in order to advance through those approval processes, it has to demonstrate benefits. And so the trials are demonstrating benefits. I think the scalability of it right now is something that I just want to see a little bit more evidence on, the lasting benefit of it. But these are questions that will be answered through the approval process and as more studies continue to be done in this area.
That's excellent. This one's actually from me, but in regards to a lot of those exciting trials that you had on one of your slides, is there any kind of sort of time frame for when things like that, particularly the one you mentioned about the stem cells in Japan, that sort of piqued my
Yeah.
interest, for something like that to maybe become available in Australia? I know it's really hard to say with things like this, but any kind of hope on the horizon kind of time frame?
It's really difficult to say definitively because the stem cells, for example, that was really quite an accelerated approval, but it's limited and conditional, which means that there's lots of guardrails and regulations around it right now because it is very early stage. But I do know that the company behind that is now going to go for FDA approval in the US, or at least sort of preliminary FDA approval and start that process. And I would imagine their plans are to then roll that out to TGA and other regulating bodies. So I can't speak specifically about their plans for Australia, but I know that they definitely are moving on a fast pace as reflected by that accelerated approval, the fact that they're going to FDA. So sometimes the gap between those early trials and translation to the clinic isn't as long as we may fear. Because remember, this isn't something that's in early stage development in like cells in a dish. This is something that already has, some weight behind it from those early trials.
Yep. Thank you. I've just had one comment come through from Simon, which was, are you able to comment on targeted therapies like DBS? I said that I understand mainly addresses the movement-related symptoms rather than non-motor symptoms. Are there any specific therapies that target non-motor symptoms or updates that you can comment on?
So, the non-motor symptoms of Parkinson's they've just proven themselves to be resistant to therapies. So, even when it comes to things like dopamine replacement therapy and levodopa, we don't see a huge response for many of the non-motor symptoms. But as non-motor symptoms have really increased in sort of awareness. And as the field is now very openly talking about the Parkinson's iceberg and the fact that your average person with Parkinson's experiences between 8 and 10 different non-motor symptoms, those conversations are what spark investment in research. So, for example, NRF has been investing in our research focused on cognitive therapies for Parkinson's disease. So we're just about to report some preliminary positive benefits from our NeuroOrb trial with Dave Hobbs at Flinders. And NRF has funded the next stage of that work to pair our NeuroOrb stimulation with stimulation of the prefrontal cortex to see if we can increase the benefits. So that's an example of a non-motor therapy. We're currently developing, this is very early stage, it's preclinical, but it's a therapy aimed at pain in Parkinson's disease specifically. I know that there are a number of my colleagues who are working on things like vision in Parkinson's disease and looking at some of the visual changes we see in Parkinson's. So there are definitely things coming, but many of them are non-pharmacological just because the pharmacology of treating these with like a pill, for example, has just proven a little bit challenging.
Thank you for that. Um, One more question, just in regards to you mentioned the different subtypes in the beginning, so how there was that sort of gut related. And
Mhm.
just if there's any research looking into things like probiotics or even the fecal microbiota transplant, where we're trying to implant sort of that healthy gut bacteria into someone who might have something like Parkinson's.
Yeah, there are a lot of studies that are starting to sort of move in that direction. There's a lot of interest in the microbiome in Parkinson's disease and in things like the FBT transplants. So I do think we'll see more on that come out. It'll just be a question of sort of how representative is that to the disease as a whole, given that in idiopathic Parkinson's disease, it seems like two-thirds of the cases are really more that brain first Parkinson's. So remember that brain body first is still very much still a hypothesis. They're still trying to confirm it.
Thank you. Um, So I think we've managed to get through all the questions that have been sent through. If anyone has anything last minute, please just raise your hand or pop it in the chat or Q&A. We stayed pretty close to time. But yes, I did want to say if you have any questions or you feel like you go away and you think of something, please reach out and get in touch with us. Like Lindsay mentioned, if you're interested in participating in the trials that we currently have running, send us an email and we'll link you to the appropriate people. And just in general, I just wanted to give a quick plug to the NRF. So we are based in South Australia, but we're independent. So we really fund research across all of the hospitals and sort of research institutes in Adelaide. And a lot of those teams are obviously collaborating Australia-wide and internationally. So yeah, if you're interested in learning more and getting involved, please reach out. We're super proud that 100% of all of our fundraising and donations to the NRF goes directly to research. And like I mentioned earlier, because we are sort of helping our really amazing researchers get those big federal grants, it's actually sort of any little bit that gets contributed is actually amplified. So that impact is really incredible. And obviously, in this space, the more research, the better. It's really important. We're really here to help the community and sort of drive that progress along so that we can come with some better treatments, some sort of earlier diagnosis, all that sort of stuff. So yeah, please get in touch with anything. Lindsay has shared her email, like I mentioned. If for whatever reason you can't see that, just reach out to me and I'm happy to circulate. And anyone who attended, we will circulate a copy of the recording. So you will get that probably in the next couple of days once that's available and ready to go. So yeah, Lindsay, I don't know if you wanted to say anything else to wrap up, but yeah, just thank you for everyone for jumping online. I know it's dinner time and we really appreciate you all being here and hope you got something out of it, the session today.
Thanks so much for coming, and I hope that you leave tonight feeling more hopeful about where Parkinson's disease research is heading worldwide. Because I know that as a Parkinson's researcher, I'm really excited for this time and for what's ahead. And I really hope that the advances I've talked about have a fast pathway to impact for the people in the community living with Parkinson's.
Excellent. Well, thank you so much, everyone. We might leave it there. Thanks for being with us tonight.
Thank you very much.
See ya. Thank you.
Bye.